viewer. Welcome to the next session that we want to have today. Now, in the previous session, we were talking about the cell membrane, how it looks like, and how it assists in the transport. Today, I want us to look at the cell transport and the fission, what is involved in this chapter. Now, in logical scientific analysis, it is very clear for us that the membrane is able to create different concentration both in the outside and in the outside. So therefore, this one gives a membrane a clear power over controlling the substance that is getting in and the substance that is getting out. So that is one thing that is very clear for us to note. Due to the nature of the membrane, it can sustain two different concentration, that is outside and inside the cell. So that is very much worth of noting at this particular point. Now, that one has given us a clear indication that the membrane can control partially what is getting in and what is getting out. Now, apart from that, we also know that the nature of the particle that is moving here is also playing a role, whether they're going to pass in or they're going to remain where they are. This one is controlled by the sizes of the particle, how they look like, and that is one thing that is very important for us to note about the cell transport. So cell transport is controlled by the membrane and also the nature of the particle that is found in either sides. Now, some molecules can easily move freely across the membrane, like the molecules of the gases and the liquid. But we also have got some molecules that may always need energy to move across, like the ions and some specific larger molecules. So these ones here now will be helping us to really understand how the particles are going to pass across the membrane. So I want us to look at the main type of transport across the membrane. So we are supposed to look at the main types of transport. Types of transport across the membrane. Now, this is something that we have talked about in the early level of learning, and we just want to look at it into detail so that we can have a clear understanding of how we have got substance being transported into and out or around the cell. Now, this one is done by different mechanisms. So we have got two, we can separate them into two mechanisms. The first one we can refer to it as a passive transport, and then we can also have got the next one being active transport. So these are the two mechanisms that help in the movement of particles across the membrane. Now, what do we mean when we talk about the passive transport? Now, like we talk about the word passive, this particular movement here doesn't depend on the cellular energy that is coming from cellular respiration, but it will rely on the concentration gradient of the molecules. We talk about the apart from the concentration of the molecules, the energy that the molecules themselves possess, like we have learned earlier, the kinetic energy of the particles, that one will also be helping much in pushing the molecules. We talk about the pressure and electrochemical forces that is going to help us in the movement of the molecules across. So that is under the passive. So the main idea is that passive, it is called passive because it is not relying on the energy that is coming from the cellular respiration, but it has got the energy from the molecules themselves. Now, when we talk about active transport, now this one here will need energy from the cellular respiration. So it is the respiration process that will be pushing the molecules across when we are talking about the active transport. So that is the two different mechanisms that we have under the transport of molecules across the membrane. Now I want us to look at this particular passive here. What are the exact examples of the passive transport that we have? Now we can clearly understand them in two perspectives. Now the first one that we want to talk about is diffusion. Now when we talk about diffusion, I think this is an idea that we already have, and we can clearly separate it. We can advance it a little at this particular point. So when we talk about diffusion, we mean that this is the movement of particle in a liquid or in a gas down across the concentration gradient. So when the molecules are moving in a liquid medium or in the gas, and it is going along the concentration gradient, we will be talking about diffusion. So this one can take place freely across the membrane. Like for example, when we talk about the movement of the gases like oxygen, it is not necessarily controlled by, interfered with the barriers like you have seen the barriers that we have in the cell membrane. So it can freely take place across the membrane, either with phospholipid layer of the membrane or in the channels of the protein that we looked at the other time. So it is not controlled by any barrier. So it takes place across freely. The movement of the carbon dioxide. So those are cases whereby we talk about the diffusion. Then we also have got another form of movement in the passive transport, what we are calling facilitated diffusion. Facilitated diffusion. Now, what is the difference between facilitated diffusion and the diffusion that I've talked about earlier? When we talk about the facilitated diffusion here, this one takes place through the carriers. So we have got the molecules that they don't rely on the energy from the cell, but they need the carriers, like we have with the protein carriers that will be passing them across the membrane. So that is the difference. So they'll be following specific channels. So these channels, we can be actually calling them the gated channel, whereby the molecules are allowed to pass through when these channels open up for them. Because the membrane has got a barrier, it can control what is coming in. But we have got specific molecules that can only be carried by specific carriers. So the specific carrier will be carrying a molecule across the membrane, and therefore they are coming from where they are more to where they are less. So that one is actually a facilitated diffusion, because it is depending on some carrier or some molecules to pick the substance across the membrane. So these are still passive transport, but they are two different clear ones. So these are example of the passive transport. Now, apart from this, we can also have got the specific passive transport, which we can call osmosis. Now, osmosis again is not a new term, but we can call it a special or a specialized form of diffusion, whereby we have got the movement of solvent molecule across a membrane of a cell. So it can be coming into the cell or it can be going out of the cell, depending on the potential of the solvent molecules that we are referring to in this particular case. So here we know that it is going to pass through a semi-permeable membrane, like we have been talking about, because the membrane can accumulate different substances across it, because it is always regulating what is passing in. So it is going to give us a scenario whereby if any case there exist solvent molecule at different ends, and then they have got different potential, there is likely of movement of particles. And again, this one is going to take place across the gated channels, like the protein channels that we looked at in the cell membrane, due to the fact that there are some particular section of the membrane that will be acting as a barrier that we have seen the prior. So this is how the passive transport looks like, and this is how it operates. So it is very clear, or it should be worth noting, that when we talk about diffusion, we are talking about the movement of the gaseous molecules across the membrane. Facilitated diffusion, we were referring to the specific carrier at least lifting the molecules from one end to the other. And then osmosis, we are talking about the clear solvent, which of course in the living organisms we majorly talk about the water molecules. So that is one thing that we should be noting under when you're talking about the passive transport. Then we also have got the active transport. The active transport. So let us see this mechanism. How does it work? Or what are the factors that we should be noting here? So we have got the active transport. So active transport mechanism. Now the thing that we noted prior is that when we are talking about the active transport, this is a type of movement of molecules that is facilitated by the energy that is coming from the cellular respiration. That is the cells carrying out respiration to generate energy that is needed to move the molecule across it. And in this particular mechanism here, we need two clear things. Number one thing that we need here is energy requirement here, requirement for this mechanism. For the mechanism, we have said one is energy, and the source of energy here is cellular respiration, which is actually given out by the molecules that we are referring to, adenosine triphosphate, ATP. So this is the main source of energy. And then we also need the carrier protein. This one will be binding with the molecules and at least moving them across. So this particular carrier, they are always specific to the molecules that they are going to transport, because they have got different shapes that can collect the particle that is going to be passed across. Now, apart from the carrier and the energy, this movement normally goes against the concentration gradient. So the movement of molecule goes against the concentration gradient. So this is how this particular molecule, how the mechanism normally works. So here we talk about the carrying of the molecules, like the molecules of the ions. Like specifically we have got the ions like sodium ions, potassium ion. This one we have seen during the impulse transmission. So this particular ion must be transmitted or must be passed across the membrane so that the next impulse is transported. So during that particular process, we really need active transport to really help us in moving these molecules. Now, this particular active transport mechanism, we can further identify some two clear processes that also depend on the active transport mechanism, like what we are calling, we have what is called endocytosis, and we also have got what is called exocytosis. Now, how do these particular two processes work? Now, when we talk about endocytosis, these are the movement of large molecules, is a movement of large molecules through cellular vesicles into the cell. So when we talk about the molecules that are outside the cell, and then they are large in case, they can be carried in a vesicle. So the vesicle can be existing, they can form vesicle, and then they move into the cell. And then when it is happening that they are forming vesicles so that they move into the cell, they move through what's called endocytosis. So moving into the cell through formation of the vesicle that is existing on the membrane of the cell that enable the cell to acquire more materials like the fluid, which are fluid in nature, into the cell. Then when we talk about exocytosis, again, this one is when the vesicles are existing in the membrane, inside the cell, but next to the membrane of the cell, so we can allow the molecules to be released outside the cell. This one happens in what example like the steroid hormones. So you have got the movement of particles, larger particles for that matter, larger particles from the cells through vesicles which exist next to the membrane of the cell. So the vesicles will be enabling cells to be releasing materials that they have, or organelles, for example, like we have got the Golgi bodies that is dealing with a lot of packaging and the releasing of the materials. So the process that they rely on in this particular case is exocytosis, and these two processes are active transport because they rely on energy that is coming from cellular respiration. So this is the main type of transport that we have in the cell. So to summarize down, we have talked about the passive form and the active form. And the passive, we have looked at it in terms of what is involved. Like we saw diffusion being involved. We saw the process like osmosis and facilitated diffusion, and in each we saw what we were talking about. And then we also went to the active transport, whereby we were talking about these processes are involving the movement of particle, but in the process we are talking about the need of cellular respiration. So there is energy, and the main form of energy in the cell is ATP. We have talked about the involvement of the carriers that will be binding with the particle to be passed across the membrane. And then we also talk about this movement is actually going against the concentration gradient. And we have talked about the need of this. Like for example, we need this process in terms of when we talk about the impulse transmission, whereby we have got the ions like sodium ion and potassium ions moving across the membrane. And these particular ions, we say that they are moving across a specific channel. So we are talking about the gated channel. So this channel will only be opening up for a specific molecule to pass because of their sizes and their nature. So that is where we need them. Then we talk about some processes that can always help in the cell functioning that rely on the active transport. Like one of them, we talk about endocytosis, and here we talk about the movement of the fluid molecule, which are large in size, from the outside cell into the inside through cellular vesicle formation. So this one will be enabling the particles to get into the cell. And then we also talk about the exocytosis, which is also the movement of particles, large particles from the cells, due to the fact that we have got the vesicles that exist in on the membrane of the cell or in the organelles. So this one enable cells to secrete materials. Like an example, we have talked about the Golgi apparatus that normally deal with a lot of secretion and also the release of the particles. So that is how this particular mode of transport really works. We are going to talk about singling out different mode of transport. We want to start from the diffusion. So diffusion, in physical term, we talked about the movement of molecules from the region of their high concentration to the region of lower concentration. So when we talk about the movement, is the movement of particles of gas or liquid from their region of high concentration to region of low concentration. So that is the simplest definition that we could give for it. Now, when we talk about this, we now know that the molecules will be moving from where they are more to where they are less. Now, let us understand clearly what makes the molecules move. Like in the previous lesson, I told you that in terms of this particular movement, this is a passive movement. So the energy that we are talking about is not originated from the cell, but it is the particle. So how do particles move? In the previous lessons, we said that the particles themselves, they possess energy. They have energy. And that energy, we refer to it as kinetic energy. So this energy enable them to be in continuous random motion. So they'll be likely to move. They are likely to move from where they are more to where they are less because of the energy that they are possessing. So that is one thing. Then number two, we said that the molecules, the membrane can be regulating the particle that is passing because of the barrier that exists due to the nature of the particle that is moving. Now, there are also other factors that will be affecting the movement of these particular molecules. Like we have said, energy here that the molecule possesses is a factor here. Now, temperature is a factor because temperature can affect that particular energy. So temperature here can affect the movement of the molecules here. This is another thing that comes in, temperature, which affect the energy, that the particle have. This is a factor that will be affecting the rate at which these particular molecules are moving. Now, if this is the case that we have here, we therefore know, if we have an example like we can have a solution here. Inside it I can have, let these circles here act as the particles, the solvent molecules. So I can call them water molecule, and then at this point here I can have these small dots here to represent for me potassium in water. We can talk about potassium manganate 7 in water. So we know that these molecules, if you look at them, they are highly concentrated at this particular point. So these molecules can move by diffusion. So how will they move? They will tend to move to areas where they are few so that they can spread in this particular water. So at the end of the day, we'll be having this particular water of mine looking like this. It will be having this kind of appearance if diffusion has taken place in it. So we'll be seeing that the potassium manganate 7 will spread in this until this spread will remain uniform in water. Now maybe we can develop an argument, because molecules normally move from where they are few to where they are less. Once they have spread evenly in this particular water like we are seeing here, can we say that the movement of this particle stops? That one will not be true. They will keep on moving. The only difference is that at this particular point there is no gradient that is lying for them to move in a specific direction. They are evenly distributed, so they are moving in equal direction that will be maintaining the uniform mixture that we are having in this particular solution. So that is what has happened at this particular point. So molecules will keep on moving because they have got energy, but reaches a point whereby the gradient is not existing because they have uniformly spread across the medium that they were traveling across. So the movement will remain in specific all direction because we have reached that particular equal distribution of the molecule across the membrane. So this is the picture that we are having. So this one will be controlled at that point. Then if we look into detail, how about the facilitated diffusion? How does it happen? How does these molecules move across the membrane using this particular mechanism? So let us see how this one works out for us. So when we have got a facilitated diffusion, under this we had agreed initially that here we have got the specific molecule that are going to pass across, and then we have got the carrier. So we know that some substances can have charges on them. They can be strong positively charged or strong negatively charged, and then they can be large molecule, and therefore they cannot just pass across the membrane of a cell because the membrane is partially permeable. So do we still need these particular molecules to pass? Yes, we need them because the cell needs to function, to keep on functioning, and there are very many molecules that are needed for this. These molecules must be acquired. So how will this really happen so that the cell can have these molecules moving across? So we have said that the cell will be having on the membrane, we will be having some specific channel that can allow specific molecules to pass across, and we are referred to them the gated channel, if you can remember the gated channel. So we call them just like the normal gate. You will only open the gate when you want to get in and when to get out. So this gate will be opening for the specific molecules so that they can get in. So we also call them the protein channels, like we study in the cell, protein channels. So we can refer to them. So this particular wording here is very much important to us when we are referring to the facilitated diffusion here. So these particular molecules, how do they move through these particular channels? Now these channels here, on those particular specific channels, like if I refer you to the membrane, the early cell membrane, remember the phospholipid layer, and we also have got this one here. This is how the cell membrane may appear. I hope this structure you are familiar with it up to now. So this is how it looks like. So when we are talking about the facilitated movement, along this particular membrane we can have a carrier, what we are referred to is as a carrier. And I want you to remember that this carrier has got specific shape which can only allow binding of specific molecule. So that shape can allow it to bind to a specific molecule, like let's see the shape that I have indicated here. So you see there is a shape in here. So if I have got the molecules that are highly concentrated here and they need to pass, how will that one happen? So if, for example, this one become amino acid, the molecule of amino acid, and the cell need them for various functions that up to now I think we are aware of. So they want to pass to this particular end. What will happen? This particular molecule, since the carrier here, this is the carrier protein, is having a specific shape for the specific amino acid. This amino acid will get in here, in that particular shape. And then what happened? Now, this particular carrier here will keep on changing the shape so that it turns over and carry this one across to the other end. So we are seeing it going to go in this direction. How will it happen? We are going to see that when we come to, this is at the beginning and towards the end, so that the molecule shall have been transported, what picture will we see at the end of the day? So this is what we are going to have when the molecule is being transported across. So now I want you to be very keen and see how the shape is changing so that the molecule is transported. So at this point here, we have the other part locking up. So this is what we have. This other part here is now looking like this. And then this is now the shape that we are having here. This part is locked up, and then we have got the molecule being released down here. So you see, this is now where we have got the molecule that we had initially up here being changed here, and then it moves and then it is being released out. So it has been transported across the membrane. So this one will continue until all the molecules here of amino acid shall have been passed to the other end. So this carrier here is very important for us. Keep on changing the shape to accommodate the new molecules to be transported until all of them shall have been transported or carried across the membrane. So here there must be an first existence of the gradient. Where is the gradient? We have got the molecules highly concentrated at one end. They are few here. So that one creates an existence of the concentration gradient. And then there is a carrier here that will be like a facilitator. It makes it easy for these molecules to move. And I've said that the carrier here is of specific shape to specific particle that is being carried across. So when it is amino acid here, this amino acid must be accommodated in this particular size here. If it is a sodium ion, they must be of the shape that can only carry the sodium ion. So they are specific and they are limiting it. So they are passing through the channel here that they are being passed across. So that is how the facilitated diffusion is taking place. This is how it actually acts. And if we look at the cell has got various portions. If we draw it into the entire cell, how the shape will be looking at, or how it's going to be working, we can have a summary of the transport of molecules like this. If I have got if this is the cell membrane, which clearly up to now you understand. Remember the water hating part and the water loving part. So this is a section of the cell. So what can easily pass here? Remember, this is where we have got the barrier. So the solvent, the water molecule, will not easily be passing here, because there is this particular hydrophobic part that will not allow water to pass in. So what will happen? Here we'll be having the molecules like the gas can easily pass here. So we can have oxygen. If it was oxygen, it can be diffusing at this particular end, or carbon dioxide. So at this section here, these ones are not having a barrier to stop them, so they'll be moving. At this other section of the cell, we can be having the channels, the protein channels that we talked about, that will be allowing the water molecule to be passing across. So which process can be passing across this particular end, or can be occurring at this particular side of the cell? Since this one is a channel that will be allowed the solvent molecule to move, so we can be having water molecule moving at this end. Therefore, osmosis can be taking place at this particular section of the cell. That one does not mean that the other molecules are absent. They are here, but there is a channel there for the water molecule to pass across. So this is what we are having at this section of the cell. And then we can also have got other facilitated movement happening across the same cell here. So here, remember, we have talked about diffusion, and then here we have talked about osmosis with the channel here, and then we can also have got the facilitated diffusion, whereby at this side of the cell we can be having the presence of the carrier here. So we can have a specific carrier that have got a specific shape. So I can draw it here for you like this. So this is the shape we are referring to. So the molecules can bind to this particular end, and then it turns so that it changes the shape and move with it, so that the molecule is passing across. So this one we can say is a facilitated diffusion. This is osmosis, and then this is diffusion. So that is the summary of the passive movement of molecules across the membrane. I want us to understand a concept here about osmosis. Now again, osmosis is not a very new idea to you, but I want us to advance it so that we understand what terms are we referring to when we are talking about osmosis at this particular level. So basically, osmosis, we can refer to it as osmosis in a cell is defined as the movement of free water molecule through a partially permeable membrane down a water potential gradient. Now I want you to be keen with that. We say that is the movement, osmosis is the movement of a water molecule, and this one is happening across a partially permeable membrane, across partially permeable membrane, and this water is moving down a potential gradient, down potential gradient. So that is what we are calling osmosis. Now there is this particular word that is coming out here that I want us to understand, a down potential gradient and the water potential area. Now what do we mean when we talk about the water potential? What are we really talking about? Water potential. Now water potential of solution simply means that when we have got a concentration, I mean different solvent, different solutions, like we can have in a cell we can have a solution. Now how is solution made in the cell? So in the cell we have got water here, is the solvent here. Now we also have got molecules in the cell, like we can have simple food molecules like glucose. We also have got the plasma proteins here, plasma proteins like fibrinogen, like for example. Now looking at these particular substances, they can dissolve in water, so they can form a solution in the cell. We also have got in this case we can have that the water molecule here, the solvent, they can be more. So in the event that we have got more water molecule, this water, that is the extra one, the extra water molecule, when they are more, they become free. So that one means that they can move. So when they are moving to an area that slightly the water molecules are not free. So at that point here, we are seeing two solutions that are existing. So when there's two solutions are existing, and then we see that there is that difference in the numbers of the free water molecule, then a gradient is created. So that gradient is what we're calling the potential gradient. So it is a gradient which can enable water to move from where they are less to where they are few. So this word potential gradient is always going to be our reference point when you are defining the term osmosis. And then it is coming from where the water has got the potential. They are free to move, and they are going to where they are actually fixed. So that is really important for us to note at this particular point here. It's something that we are going to talk about more. So the potential gradient is existing when we have got two solutions that are having different concentration, and this concentration will be based like the cell, what we have in the cell, like the food molecules, the existence of the proteins that can dissolve. But remember that these particular molecules that are existing in the cell, some cannot affect the movement of water. Like we have got starch here, which is not soluble, and therefore it cannot affect. We have got lipids. These are large molecules, and then they are not soluble. Therefore, they cannot affect the osmosis of the cell. They cannot change the concentration of the cell. Therefore, they are not going to be. We are not going to use them here to influence the osmosis that we are talking about. But we are going to talk about the soluble molecules that are enabling molecules to move across. Like for example, if I have got a membrane that looks like this, for example, I can have the solvent. This is the water molecule. I want us to look at this illustration. So I have got, this is water molecule, and then I can also have got on the other end here, I also have got solution. So what do I see here? Now we can have the solvent and the solute themselves existing. That is what we have here, and then this is what we have in this particular membrane. This is what we have. So this, assuming that this is a partially permeable membrane, what will we see? We want to see how the osmosis is going to take place. So I have said that this is water molecule, and then these cycles of mine are, we call them the solute molecule. So looking at them keenly, we said that there should be existing a potential that is existing. So I look at it and I see that there is a lot of solvent molecule here. as compared to this. So what type of solution am I really referring to in this particular case? This one here is a solution that has got a lot of solute. So the water here, they are more of fixed here. If I look at this particular solvent here, they are few. And then the water molecules here, there are many. So we are seeing that the potential is high that they are going to move because they are free. So they can move by osmosis. Osmosis can take place here. So what is going to move here is the water molecule. They are moving. So osmosis is going to take place. So you see, this is a very special diffusion. Why? Molecules are moving from where they are more. But it is special in that the molecule that is moving here is specifically water molecule. This solvent here, they are the one moving. So the solute, they'll remain behind because there is a barrier that they cannot pass through. So these molecules will be moving at this particular end. So that is what is happening in terms of the osmosis on the cell. That is how it happens. Now, these particular molecules, the movement should be regulated, especially in the animal cell. Why is it so? Now, this movement here, if any case it happens unregulated, we will see the case whereby the cells will be expanding, they'll swell, and eventually they can burst, especially in the animal cell, and therefore this one can make various processes to stop, or the cells can really lose a lot of water until they shrink. The shape can change, and then the activities of the cells can stop. So it has to be regulated so that the cells remain alive, so that they keep on functioning as required. So this osmosis here, actually it should be really regulated in the cells. Although the plant cells here cannot actually burst based on the structure that they have, the cell wall, but again, just like what we have seen in animal cell, in the event that we lose too much water, there will be no medium for the reaction. Remember, the water in the cell forms the medium for reactions and the transport. The moment any cell is losing too much of it, or almost all of it, there will be no medium for the reaction. And examples of the reaction that we talk about in the cells are those ones that will be either creating the new molecules like the protein, or providing the energy. And when that one stops, that cell will definitely die. Therefore, even if the cell of the plant cannot burst, but it should be regulated for that reason, that the activity of the cell must be maintained, and we need the fluid inside the cell to help us in doing that. So we have to actually maintain the process here has to be regulated. So that is how it happens. We can have a model here to explain it. We can have another example here, a model here that can be seen physically, and we can actually explain it. So suppose we have got a setup that looks like this. We can have the artificial permeable membrane, like we can have the whisking tubing, which can be helping us in regulating which molecule is moving across. So you can have a tube here, and then we tie the partially permeable membrane here, and then we put it in a water container. So this is what we have. So I want to have two molecules here, the molecules of water, like this one. You can immerse it in water completely, and then inside it we can have another solution here, so that we have got the potential of water existing between them. So we can have the sucrose solution. So I want to indicate them. So this one becomes sucrose solution, and then this is water. So you can see that there are also water molecules here, but few. So there exists a potential difference here. So we have got water molecule that is outside the whisking tubing. This is whisking tubing. It is partially permeable. So what will happen with time? Now we will see the case whereby the permeable membrane will allow water to move in, because this water are free, so they can move, and then they get in. Eventually what happens is the volume here will increase, and we can see that water is flowing through the tube up here. So in the final diagram that will be illustrating the happening here is this. I will be having now the volume here really expanding. So this is still our whisking tubing. This is water, and then the water now molecules getting in, and you're seeing a case whereby the level will rise so that it gets more space for the water to occupy. So this is what we are going to see happening here. So this is something that can be demonstrating for us the actual osmosis and how it happens across the cell. So this is what we have that we should be understanding. So I hope we can remember that there is osmotic concentrations, that is solutions of different nature that will be enabling the water to move in. Like if I have a case whereby I have got two solutions that are of the same concentration, such solutions we are going to refer to them as isotonic. These are two solutions with equal concentration. So the water molecules, they are balancing on each side. So in this case, there is no osmotic potential, there is no movement of water across this particular membrane so that they can mix within one another because they are having equal concentration. Then we also have got hypertonic solution. Hypertonic solution. So in the hypertonic solution here, we mean that the osmotic concentration of solution here is lower than the cytoplasm, for example, if we are referring to them with the cytoplasm. So in this case here, the cytoplasm is having a lot of solute as compared to the surrounding. So the surrounding here has got free water. So in this case, if this solution exists between a surrounding and a cell, the cell will definitely gain water because the cell has got a lower potential. It has got a lot of solute, and therefore water can easily move in. That cell will gain water and increase in size. Then we also have got the hypertonic. So in the hypertonic solution, we mean that we are talking about the osmotic concentration of the solution that is outside as compared to the one that of the cytoplasm inside the cell being now low. So the cell here will be losing water to the surrounding. Now these ones are very much important for us, so they help us to see how the cell will be functioning, so that we see which cell will be gaining water and at what point, and which cells is not going to gain water, and how will this one be helping the cells. So this one can be helping us to understand how osmotic take place in the plant cell and in the animal cell, and how is this of importance to us. So if we talk about osmosis in animal cell, now we have said that animal cell can gain water, equally they can also lose water, and this cell, especially the animal cell, is very fragile. Why is it fragile? It does not have that particular hard membrane that can stop the gaining of more water, and therefore it can reach a point where it extends and then it actually bursts, or it can lose excess water until there is no medium for the reaction in it, and therefore it makes the functioning of the cells to really stop. So how is this really important for us to actually worth noting? Now these particular cells, like when we talk about a cell like let's say red blood cell here, it's a very important cell. We know that the function of this cell is very vital in the transport of the gases. Now this particular cell will therefore have a substance that can carry the molecules like let's say glucose, which will now be helping us in regulating the osmosis that is taking place in it. So it has got a special carrier. The cell has got a special carrier that can be carrying the glucose, so that at least it can be using this glucose for energy, yes, and also regulating the osmosis that is taking place between it and its surrounding. So this particular osmosis in this cell, especially this one here, becomes very vital for us to understand. So that is one thing. So it is good that this cell maintain its disc shape that we know of. And this shape will only be maintained if the osmosis that is taking place, the osmotic potential of this cell, is always correct, so that it doesn't gain too much water that can make it to burst, or it can, that is, it can really expand until the function stop, that is gaining of water, or it can be losing water until the shape now changes until it stops functioning. So we know that the correct shape should always be the disc shape. So that is very important for us to note that. That is very vital for us to know that. Now, equally in plants, how does this one really work, or how do we retain it so that we don't have a case whereby there is no activities that are taking place in the plant cell? Now, I want you to note that when osmosis is taking place in the plant cell can gain water, especially when the cytoplasm, you can talk about osmosis in plant cell. The plant cell can gain water. That is whenever the sap of the cell is hypertonic to the surrounding. So there is that particular hydrostatic pressure that will be created. The cell will be gaining water. So we have got a hydrostatic pressure going to be created. And what do you mean here? This is the case whereby the cell is absorbing water. The cell is absorbing water. So when it is absorbing water, what do we know? We know that the vacuole is going to expand. And that expansion of the vacuole is going to create a pressure which is going to pull the membrane towards the wall. Membrane is pulled towards the wall. So that particular pressure is hydrostatic pressure. These ones make the cell to remain in a certain shape, which is very hard. So we call it a tagger. The cell become turgid. The cell become turgid, very, very hard. That is, it has absorbed too much water. So at this particular point here, there will be the pressure that is going to be acting on the wall due to the pushing of the membrane towards the wall. Now that cell wall will not just hold back. It will also come to resist this particular pressure that is coming towards it. So when it is resisting this particular pressure that is in it, it is developing what we are calling a tagger pressure that resists the hydrostatic pressure that enables the cell to have the definite shape that it needs for it to function. So this is a pressure that is developed by the wall, developed by the cell wall to withstand the force that is acting on it. So that is how the cell is going to really work towards this particular functioning, so that it doesn't gain too much water, necessary water that is not needed in the cell. So that is one thing that's going to happen. Equally, the cells can also lose water, and when they are losing water, the shape will change. The shape will change, and then we will say that the cell will become small and become plasmolized, because they have lost the water content. So they can lose water, and become plasmolized. So it is very important for these processes to be regulated in the cell, so that the cell have got a clear functioning shape that they need to retain all the time. So that is what we should be understanding by osmosis as a special case of diffusion. Thank you.